accreditation for higher education. And it's also accredited by Accreditation Agency for study programs in engineering, informatics, natural science, and mathematics. The Master in Fisheries Science has collaborated with several universities abroad and has been awarded a double degree with the University of Tasmania in Australia. This master program has also carried out program development led by Erasmus Mundus with the European Union. The study program has been accredited excellent accreditation by the National Accreditation Board for Higher Education. The Doctor in Agricultural Science has a vision of being a pioneer at the national level and internationally in producing doctors who are able to develop science and technology in the fields of agriculture and fisheries, serving the interests of the nation and humanity based on Pancasila. The department organized a doctoral program for those with an interest in fisheries and marine science. His study program has been accredited A by the National Accreditation Board for Higher Education.
The Department of Fisheries has been involved in carrying out of campus learning through the program of Merdeka Belajar, Campus Merdeka, Learning Independent Campus, or MBKM. The MBKM policy is implemented to realize an autonomous and flexible learning process in higher education to create innovative learning in accordance with student needs and to improve employability. Currently, the Department of Fisheries has implemented the MBKM program in the core of entrepreneurship, internship, student exchange, community service, and research.
In the implementation of this program, the department collaborated with several governments and companies, such as PT Suritani Pemuka, PT Multidaya Teknologi Nusantara or e-Fishery, PPN Perigi, SKIPM Yogyakarta Kelas 1, PT Sarana Tani Pratama Jembrana, PT Alga Bioteknologi Indonesia Semarang, and others. Some activities in fisheries include classroom activities, practicums, and field practicums. E-learning activities is equipped with complete and adequate facilities. The Laboratory of Aquaculture has two sub-laboratories, namely the Fish Breeding and Hatchery Laboratory and the Fish Nutrition Laboratory. The Laboratory of Fish and Environmental Health Management is in charge of the Hydrobiology Sub-Laboratory. The Laboratory of Aquatic Research Management consists of two sub-laboratories, the Aquatic Ecology Laboratory and Fisheries Research Management Laboratory. The Laboratory of Fisheries Socioeconomic and Fishing Technique. The Laboratory of Fish Processing Technology. And the Laboratory of Fisheries Product Quality and Safety. Research students can also use the Agrotropical Learning Center to do their research in the Fish Biotechnology Laboratory and the Marine and Terrestrial Venture Laboratory. In addition, there is EMB, or Business Incubator, which provides a forum for educating and developing the entrepreneurial spirit of students. The department also has a library, study room, examination rooms, and podcast room that are comfortable and clean, which can be accessed by all fisheries students. It's been 58 years. The Department of Fisheries has contributed to advance Indonesian marine and fisheries affairs with various activities, such as routine scientific activities on a national event, the National Seminar on Fisheries and Marine Research or SEMNASCAN UGM, and international events through the International Symposium on Marine and Fisheries Research, or ISNFR. In addition, the department also actively conducts training related to fisheries and marine affairs, such as fisheries product processing and HACCP training, aquaculture training, and conservation training. Apart from academic activities, students and lecturers are involved in community development activities in accordance with the principle of the Tridharma Perguruan Tinggi. One of these activities is counseling, assistance for micro, small, and medium enterprises and water conservation. The department has received various achievements in various fields, including first place in the female open class of the Gupi Aqua Fest IPB 2021 National Contest, Grand Prize Award International Student Video Contest 2021, Best Top 50 Young Leader in Festival Kaum Muda untuk Iklim dan Kemanusiaan 2021, third place in Poster Competition 2021, first place in National Scientific Paper Agriculture Farming System Competition, first place in Scientific Article Airlangga Nursing Journalist Festival 2021, first place in the National Research and Development Competition, and the others. Not only achievement by students, the lecturers also made achievement in various prestigious national and international events, including creation of patents, publication of international journals of international reputation, and producing various other works. These are students' organizations for skills and talent development, such as diving units and fisheries students association. Salam Perikanan UGM is a unit for fisheries students to develop their organization and diving skills. Established on October 8, 2018, the unit continues to create license skills for graduates, especially the A1, or Open Water Diver OWD CMAS 1-star license. KMIP or Keluarga Mahasiswa Ilmu Perikanan is a student association of the Department of Fisheries, which was established on 19 December 1977. KMIP acts as a forum that facilitates the activities of all fisheries students in developing insight, soft skills, personality, and professional interests. In developing professional interests, KMIP oversees the Ornamental Fish and Aqua Escape Study Group or IHA and the Coastal Community Service or CCS, a study group that examines the culture and social life of coastal communities. Until now, the department is still developing human research, knowledge, and technology for Indonesian marine and fisheries. Let's together with the Department of Fisheries in Universitas Gadjah Mada, we can create the better Indonesian fisheries.
Hello, Prof. Takeuchi. How are you?
Good. How are you? Hey, thank you.
Yeah, nice to meet you again.
Very good.
Now in Japan it's summer, right?
Yes. Summer, very hot.
Very hot.
Yes. We just almost finished the rainy season.
Oh yeah, I know. Yeah, usually in June it's hot rainy season.
Yeah. So you will give our opening speech.
Yeah.
That I
So
will.
we will start, Dr. Rini. Or
Mbak Sulis yang akan
Sulis, yeah. You may start.
Yes, Prof.
Yeah, that's why you need to only. Okay, yeah.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to SinnTech Webinar 17, the Advanced Technology of Fish Breeding. My name is Setyawati. I am honored to be Master of Ceremony for today's event. We have an exciting and informative program lined up for you today, and we hope you will find it both enjoyable and enriching. First of all, I would like to express my respect. The Honorable, the Head of Fisheries Department, Professor Dr. Alim Isnan Setio, Master of Science, the Honorable Professor Yutaka Takeuchi, and Dr. Priadi Setyawan, the Honorable to all participants in this webinar. To begin, I would like to invite Prof. Alim Isnan Setio for opening speech. Ladies and gentlemen, please join me in welcoming Prof. Alim Isnan Setio.
Okay, thank you very much. Good afternoon, everyone. My Excellencies, Professor Yutaka Takeuchi from Kanazawa University, Japan, and Dr. Priadi Setyawan from National Research and Innovation Agency Indonesia, and all participants in this SinnTech webinar series. Today is a good opportunity, and it is an invaluable opportunity as we will learn about the advanced technology of fish breeding, which two distinguished researchers, Professor Takeuchi and also Dr. Priadi Setyawan. Thank you very much, Prof. Takeuchi, and also Dr. Priadi Setyawan for your time to give lecture and share your long experience in the fish breeding technology. Today, SinnTech is 17 SinnTech, and we commit to conduct this webinar periodically. So we invite university, research institute, and also private company to collaborate for the next SinnTech event. We hope that this SinnTech become an effective event to share knowledge and experience among us. The last, I officially open this 17 SinnTech. Thank you very much.
Thank you very much for your speech, Prof. Alim Isnan Setio. Yeah, ladies and gentlemen, now we move to the main part of our webinar, the presentation from the speakers. Our esteemed speakers today are Prof. Yutaka Takeuchi and Dr. Priadi Setyawan, who will be presenting on the advanced technology of fish breeding. And then our esteemed moderator today is Dr. Dini Wahyu Kartikasari, the lecturer of Fisheries Department, Universitas Gadjah Mada. Give me a warm welcome to Dr. Dini to moderating this webinar.
Thank you so much, Mbak Sulis, for the time. Good afternoon, everybody. Good afternoon, Prof. Takeuchi Sensei, and also Dr. Priadi. It is our pleasure to welcome you. And I'm Dini, will be your moderator today. Before we start, let me introduce Prof. Takeuchi Sensei first. Okay. Professor Yutaka Takeuchi is a professor in Faculty of Biological Sciences and Technology, Kanazawa University. He is completing a PhD from Tokyo University of Fisheries on 2002, and then his academic specialty is molecular and cellular biology, reproductive biology, reproductive engineering, developmental biology, aquaculture, seed production, fish transgenesis, and also germ cell transplantation technique. Prof. Takeuchi has so many papers in prestigious top journal in the world like Nature, and science, finance, and genetic aquaculture, and also biology of reproduction. That is a very interesting publication. And Professor Takeuchi get some awards, so very prestigious award from the Achievement Award for Young Scientists in Fisheries Science from the Japanese Society of Fishery Science, and then also Japan Prize in Agriculture Sciences Achievement Award for Young Scientists, and also Distinguished Award for Promotion in Hirameki Tokimeki Science from Japan Society of the Promotion of Science 2014. Professor Takeuchi Sensei, now ongoing research project is about establishment of germ cell transplantation technique in marine teleos, study of germ cell development in abalone, artificial spawning induction, and seed production of tunas and bonitos, and then development of information and communication technology in land-based aquaculture system, and also assessment of biodegradability of biomass plastic in marine environment, and also their use in aquaculture and fisheries. So it is very interesting topic for Prof. Takeuchi Sensei to talk about application of surrogate broodstock technology for preservation of fish genetic resources. So I think I and we can wait to hear your presentation, Sensei. So time is yours.
Thank you. Thank you so much, Dr. Dini. Let me share my screen.
Yes, we can see clearly already.
Maybe.
Yeah, you can. Yeah, shares.
Can you see? No.
Yes.
Okay.
Clearly.
Okay. Thank you very much for very kindly introduction of me. So today, oh sorry, why I have subscript here? Just as well. Okay. So my name is Yutaka Takeuchi. Can you hear me? Still hear me?
Yes, clearly, Sensei.
Today I'd like to introduce our research about the surrogate broodstock technology. Maybe for some people it's new to hear, but this is one of the reproductive assisted technology for fish. And we developed this technique about 20 years ago when I was a PhD student. And after that, this technology is now spreading worldwide, maybe in US, EU, Asian countries, African countries, and maybe I believe some of you in Indonesia are trying to do similar, trying to develop similar technology, I guess. And now I am in Kanazawa University. Let me briefly explain about myself. As Dr. Dini introduced me, I got PhD in Tokyo University of Fisheries. And after that, I have got job in the same university, but name is changed, like TAMSAT, Tokyo University of Marine Science and Technology. And I was in one of the research station in Chiba, which is close to the sea, and I start working with marine teleost and abalone when I was in TAMSAT. And after that, I moved to Kagoshima University, Faculty of Fisheries, only for 2.5 years. After that, I moved to Kanazawa University. Kanazawa University is here. So I almost traveled half of Japan and got three university job, and now I'm finally in Kanazawa. Okay. So Kanazawa is the city like this. They're very old. We have castle and traditional Japanese park, old street, and station, Shinkansen stations like this. And we have many seafood. Please let me know when you have chance to visit Kanazawa. Okay. So today my topic is about the surrogate broodstock technology. Okay, so this word surrogate is mainly used for human or mammals. Like as you see here, a couple have some problem, have some difficulty in the pregnancy. They will ask another woman to grow their embryos in her body, surrogate mother's body, to give a birth. Okay, this is the very popular and excellent reproductive assisted technology in human and mammals, but we fish don't have like this technology. And as you know, this technology is very useful for the preservation of genetic resources and make a baby. Okay, but fish, if we think about fish is different from mammals because they are external fertilization. And in the case of human, only one embryo, two embryo is okay. But in fish, we need a large number of eggs for seed production aquaculture. So that's why we try to transplant germ cells, the origin of egg and sperm, from one species to another species to develop surrogate broodstock technology. But finally, we want to make a surrogate mother and father of fish. Okay. So this picture shows the how germ cells develop in the gonads. Gonads is ovary and the testis. Germ cell means the cells for the sex. Okay. This is the gonad, and you will see egg and sperm in the adult. But if you look at the development of these cells, you will see the origin is primordial germ cells, PZC in short. And after sex differentiation, a baby female will have oogonia, and the baby male have spermatogonia. Okay, so this germ cell, this is called germ cell lineage. We can roughly separate it two parts. First part is mitotic stage. These cells, PZC, oogonia, spermatogonia, still have self-renew potency. They can make their own copy cell. Okay, and their propagation their potential is very high. Okay, they can make their copy and they can produce their large number of cells. But after they enter the meiosis, as you know, there only one oocyte can make one egg, one spermatocyte can make 4 sperm. So these cells are not very useful for transplantation. These cells, mitotic germ cells, could be useful for transplantation as a donor cell. Okay. So this is our idea. So we first isolate these cells, PZC, oogonia, spermatogonia, from commercially variable, for example, bluefin tuna, fish, and transplant these cells, very powerful cells, their origin of egg and sperm, into the recipient fish, another fish. Okay, in this case, mackerel. Mackerel and tunas belong to the same same family. Okay, and after transplant, if these cells can survive in the body of recipient mackerel, they could produce egg or sperm from transplanted tuna cells. Okay, and okay, if they produce tuna egg or tuna sperm after mating, we will get donor-derived bluefin tuna offspring from the surrogate mackerel broodstocks. Okay, so this is important picture. So let's see why we need to transplant. Why need to use transplant into the juvenile or larvae stage? The reason is the immunity. Okay. Someone put their hands. Maybe question? Or
Pak Roba, do you have any question? Or maybe just,
just
not yet, Sensei. You
yes.
can continue, I think.
Okay.
Yeah.
Oh, yeah, you can ask me anytime. If, you know, I cannot see your face now, so I'm not sure I can. Okay. Okay, let me ask anytime, any question. Okay. Right. So the reason why we use recipient larvae for transplantation is immunity. This is the classic paper. They report about the ontogeny of the immune system of fish. And especially the marine fish, they hatch very immature stage, and they don't have, their blood cells are not well developed. They don't have their own immune system. So we want to transplant cells from other species when recipient immune system is not well developed. So we expect they can escape from their immune system and can colonize the body of a recipient. Okay, so I will show you the next. Okay, so for the transplantation, we need to label the donor cells to trace the where they go, where donor cells go. Okay, so we often use the PKH26 is a name of red dye for living cells. We can label cells with a red fluorescent color without fixing or without freezing something, that we can label live cells like this. And after after we prepare donor cells like this, in this case spermatogonia from the young tuna, and labeled like this, and the cells are then sucked up by a micropipette. Here is, you will see, maybe this is 10,000 cells in this part, and then transplant into the body cavity of mackerel. You see here. And after transplantation, you will see the red cells in the body cavity of recipient. Okay, here is a movie. I hope we can move. Sometimes don't move. Sorry, it's not moved today. But this is the cells, donor cells, and I'm about to suck up the cells by glass needle like this, and transplant into the body cavity and wait for a few weeks. In this case, 3 weeks. Okay? And after 3 weeks transplantation, we kill one of the recipient fish and open the body cavity and remove gut, and you will see the tiny gonads like this. And one pair of gonads left and right, can see here. And under the fluorescent microscope, you will see many bright red cells here. Okay. And then we remove this gonad by a very fine forceps and put it on the slide glass, and look under the microscope. You see, this is a gonad. This one is here, high magnification view after removed from the fish. And you will see red cells, 1, 2, 3, 4, 5, 6, 7, 8, many red cells in the gonad. This means transplanted cells, donor cells colonized in the recipient gonads. Okay. Even between the different species, they can colonize. Okay. So next, I'd like to show you why donor cells can colonize. Okay, here, the donor cells, the donor cells, germ cells, red cells have a very unique potency for moving, walking in with extending shoot pods. Shoot pod is a name of this structure, pod, from extending by the cell. These cells very unique. They can extend the pod and they can move around the body. Okay, and then finally find their home, which is embryonic gonads. Okay, I'll show you some movie transplantation. Okay, we first transplant red labeled cells into the body cavity of a larvae like this. Okay, and then these red cells start extend shoot pods and move like this. Okay, and after they moved in the embryonic gonads, if this fish is female, they develop to eggs. And if this fish recipient is male, they can develop to sperm. It depends, sex depends on the recipient fish. Yeah. If gonad, they can be egg. If ovary, they can be egg. If testis, they can be a sperm. Yeah. So this migration is happen in their own germ cells. It's their nature. The germ cells always find when the fish grow, germ cells differentiate and then walk to the gonad. And I find a way. This is a route for germ cell migration. So that's why I transplanted these cells, donor cells, on the same route of germ cell migration. Okay. Anyway, this is the mechanism how cells can colonize the recipient gonad like this. We only transplant into the body cavity, but they walk and colonize the gonad. Okay. So next, this is a list of marine fish we already developed the transplantation technique. In yellowtail, horse mackerel, little tuna, chub mackerel, grass puffer, and the Japanese flounder flatfish. Okay, so many species, as you see, the needles can penetrate, can inject into the body cavity, and the cells are now transferred to the one species to another species. Okay. So this is a freshwater species. We can do this transplantation in sturgeon, which this is my collaboration with Chinese researchers, and medaka, and tilapia, and zebrafish, and also not us, but other people reported they can do transplantation in carp and bittering. Okay. So this is the basic principle of germ cell transplantation in fish. The first case of this technique was reported like rainbow trout sperm were produced by mass salmon. So this is the first case fish produce functional gametes of another species in the first case in the animal. And then we named this technique as a surrogate broodstock technology in fish. So now, this was 2004. So in the last 20 years, many people, students, try to develop their own species. And this guy, 2015, we successfully produced the yellowtail sperm in a host mackerel recipient. And this guy developed tiger puffer egg and sperm in a grass puffer. You see, they are closely related species, and donor is big and take a long time to get mature, but recipient are the small and short time for the maturation. Okay. So this technique I developed when I was PhD student in Professor Yoshizaki's lab, and after I leave his lab, and students still working on this technique. And I will introduce some of their achievements. First one, they can transplant the they can use frozen germ cells as a donor. So this is a bank, frozen cell bank. If the species are extinct, we can still get cell from the bank, cryobank, and transplant and regenerate fish species. And another case, they can culture donor germ cells and transplant into the recipient fish and get a fish. Okay. The one interesting story is like this. When some student lost their fish by accident and they freeze it in a normal freezer, and one of the, I will say the crazy student, he tried to get cell from this fish and transplant, and he successfully get the fish from these frozen fish. So this is an emergency case. If your very important fish died, you can freeze, and someone who have a skill can regenerate your fish from this frozen. Okay. We have some problems here. In the case of fish, fish sperm can cryopreserve, but fish eggs cannot cryopreserve because of their large size. And also, egg have a mitochondrial gene that are inherited only from the mother. So in the case of fish, sperm bank is not enough, but egg bank cannot. So germ cell bank, frozen germ cell or cultured germ cell is only way to preserve the genetic material of females. If you have transplantation, you can regenerate egg of your fish. Okay. So next, let me introduce you the advantage of this technique in fish. So for example, if we can do this in a marine fish, efficient production of commercially valuable fish with large body size and long generation time could be produced. Egg or sperm of commercially valuable species can be produced in short time and in small space. Okay? And the next one is the conservation of endangered species and genetic diversity of artificial seeds for stock enhancement. Okay, so endangered species, you can imagine. I will explain a little bit about genetic diversity. Okay, in Japan, the flatfish is a major target species for stock enhancement, but there, if you go to the sea, if you catch the wild flatfish, they have many variations. But for the stock enhancement, they use a broodstock with less genetic diversities. So the people are working on stock enhancement. They want to make a genetically diverse fish, flatfish, but it's really difficult to have many large number of broodstock like this. So our idea is make a cocktail mix of germ cells from this variety of fish, flatfish, mix and freeze, and then transplant to a recipient farmed flatfish. Okay, so that could produce the genetically diverse juvenile production for stock enhancement. Okay, so I explained already, like transplantation with red fluorescent level cells. You can confirm transplantation is success. But the problem here is we cannot distinguish multiple donors. If here we want to mix donor cells, not only from one fish, maybe 10 fish, more than 10 fish mix transplant into one fish. And this fish could produce the diverse sperm egg. But fluorescent level is not enough to check the distinguish multiple donors because they have only one color. Okay, so next idea is a microsatellite markers like this. If genetically mixed, we will see many different alleles, but the problem is the sensitivity. If you mix cells, mixed alleles, the sensitivity is maybe, if this allele is contained less than 10%, we cannot detect. This is PCR problem. So we cannot judge if this sperm from recipient can have more diverse genetic information. So that's why for this experiment, we used PCR technique in M.Si, M.Si region of the gene, because they are very highly polymorphic. If you amplify M.Si alpha gene region, one pair of primers can distinguish over 40 haplotypes from 80 individuals. So that means if you do PCR, if you get sperm from a recipient, which transplanted many different donor cells, and you take sperm DNA and you can analyze how many different haplotypes, how many different sperm you can get from one fish. Okay, so this is MHC is very useful to analyze the diversity of sperm. Okay, so this is another story, another application of germ cell transplantation in fish. Okay, that which is making eggs from spermatogonia with XY chromosome. Okay, so this I will explain you about the in the case of pufferfish, okay? Pufferfish is sex gene system is XY like human, okay? In the testis of male, XY spermatogonia can be a spermatocyte and a sperm with X chromosome or sperm with Y chromosome. This is normal case. But we take this XY spermatogonia and transplant into the female pufferfish, recipient fish, and wait 1 year. And after 1 year, matured female recipient produce both X egg and Y egg because their donor cell is XY, have XY chromosome. After meiosis, they can be a X cell, X egg and Y egg. Okay, so and Y egg, if Y egg fertilized with Y sperm, this fish will be YY super male. Okay, so YY super male. This is tiger puffer. We use Graph's puffer because they can mature in 1 year after transplantation. And we got YY super male, but we have to wait another 3 years to get this tiger puffer mature. But after 3 years, we got sperm, old Y sperm, a monosex population of tiger puffer can be produced with this sperm from YY super male. So I need to explain why we want to do this. Okay. Is this in a pufferfish farming, fishermen want males because male have, tiger puffer female ovaries have toxins, but testes don't have toxin. We can eat, and not only edible, the price is higher. Testis is over 10,000 yen per kg, but the meat is only 2,000, 3,000 Japanese yen per kg. So tiger puffer testis is very popular and expensive food, and the fishermen want to have all male seeds, but no one, you may imagine how the hormone, usually people control sex using hormones, but last 40 years, many people tried to induce sex reverse in tiger puffer, but they couldn't. So we are the first case. This is the first case we can make our tiger puffer super male. He can produce only Y sperm. Okay. So what I want to say here is germ cell transplantation technique, surrogate broodstock technique can also use for sex control of aquaculture fish. Okay. Then I will next I will like to explain you about the bottlenecks. We are still having some difficulties in this technique. Okay. The first bottleneck is like this. Okay. After transplantation, when we use fertile recipient, they will make both donor red, here I show you in the red, donor sperm, and at the same time they produce yellow recipient sperm. They're his own sperm. In a female also, we can get egg from donor, but this fish recipient also produce egg of her own egg. So that's why this causes the mixture of donor and the recipient sperm. That is not good for us, not very interesting. So to overcome this problem, we use our transplantation to sterile recipient. Sterile means now we can get sterile fish with triploidization, hybridization, genome editing, or drugs. So, but anyway, if we use sterile recipient, this recipient cannot make their own egg sperm. They produce only donor sperm and donor egg. So today I like to show you some, sorry. I will explain later which type of sterile recipient I use for our next experiment. Okay, so the second bottleneck is a genetic relationship between donor and recipient species. Now, as I show you here, colonization, after transplantation, they colonize. Colonization happens in every combinations, but after that, they disappear when genetically distant species are used. For example, bluefin tuna to croaker, bluefin tuna to mackerel, they can colonize, but they cannot survive. This is another bottleneck we have, okay? So here is a summary of the relationship of donor and recipient combination. Okay, first, you will see if donor and say recipient are same species, you can make egg and sperm of donor species. If they are, the relationship is same genus, donor and recipient belong to same genus, you can still make egg and sperm. But if they're different genus, we cannot. So genetically relationship, I don't know the reason, but somehow they affected the successful transplantation, right? So this is a big problem, still a big problem with us. So but after a few years, we got some ideas, hint to overcome this problem. I like to show you. This is a fish I used at that time, croakers, blue drum, yellow drum, white croaker, Malawi. There are species we can find in Japan for different species. And I try to make all combination of hybrids to expect we will get hybrid sterility. For example, mule, like horse donkey hybrids can survive, but they are sterile, not fertile. No egg, no sperm, this mule can produce. So this is called hybrid sterility, and I expect it to have hybrid sterile in this combination. Okay. So only one case, one hybrid between blue drum and white croaker, we got hybrid. This fish can survive. But if you look at the gonad of this fish is here. When you compare to the normal blue drum is here. When you compare blue drum and the hybrid gonad is super small. And when you make a histological section of this hybrid gonads, you will see this looks like testes, but inside empty, nothing. Only structure we can see, but no germ cells, origin of sperm, you cannot find here. This is control. You see many sperm, the dark purple dots like this is sperm, but nothing in the hybrid gonads. So when I see this could be a very useful sterile recipient for our future germ cell transplantation because they don't have their own germ cells. They have many space in the testes. Okay. So we go to fishing and we catch a giant croaker like this. It's the biggest croaker fish in Japan. Okay, we get sperm, prepare the donor cells from this fish and transplant into the body cavity of sterile hybrid. After a few months, 3 months, 4 months, I guess, we wait and try to get sperm or egg of giant croaker from this sterile fish. Okay, so let's go. Okay, 6 months, sorry. 6 months after transplantation, of donor giant croaker, we got 90 survival fish, and two of them produced sperm like this. This is sperm head, and you will see tail, and this sperm is moving. So you, as you remember, this fish is sterile, empty, but they can produce moving sperm, and we make a fertilized with the egg, and they show some developmental potency. Okay. So after artificial fertilization, I kill this fish and check the gonad. Here you see this part is a grow, and if you make a histological section of this part, you will see spermatozoa in a sterile recipient fish. So this one this experiment shows the functional giant croaker sperm was produced in sterile recipient gonads. Okay, so here is our, let's think about the genetic relationship of this species. Okay, giant croaker is here. This is the paper from our Chinese researchers. The Argyrosomes japonicus giant croaker is here. The common ancestor and the recipient is here. They belong to different genus. I said before, if genus is different, they cannot make egg or sperm. But when we use sterile hybrid, parents are here and here. Okay. Their common ancestors separate, like how many years ago, I forget. Like, okay, anyway, they belong to different genus, 18 million years, which is more farther than human and the chimpanzee. Human and chimpanzee is only 6 million years, but these two species have 18 million years ago, they separate anyway. So, but still, this fish, sterile hybrid, can make sperm of giant croaker. Okay. So here is this. We still have problems, but we showed if when we use sterile fish as a recipient, we can produce donor of belong to different genus. Okay. So sterile hybrid, when we are interested in germ cell transplantation technique, you should consider about sterile hybrid recipients because they will be a breakthrough for the surrogate broodstock technology in fish. Okay. So let me explain one more story like this. This is a picture I showed. Oh, I already almost finished my transplantation experiments in juvenile. Okay, but now I'm going to talk about the transplantation to adults. Okay, so when I look, I already show you this picture, no sperm in the testis, empty testis of sterile hybrid fish. When I first look this picture, I remember very well about the paper in the mouse. 1994, the BRIN published, find the very unique sterile mouse, testis of c-KIT mutant mouse. This mouse have a structure of testis, but nothing inside, no sperm. Oh, when I see this, the picture on the left, I said, oh, this testis looks like c-KIT mutant mouse. And this mouse is used for a transplantation experiment. Okay. When the Brinster, these guys, find this mouse, they use this mouse as a recipient of cultured or normal spermatogonia from donor species and transplant into the mutant. Okay, because this mutant mouse is empty testis, they can transplant spermatogonia of another mouse. Okay, this is classic germ cell transplantation in mammals. Okay, then they don't use embryos because their embryo grow in the mother's uterus, so they cannot access to. They cannot transplantation when the mouse is young embryo, but they can do adult. Okay, so because our fish looks like this mouse, so we want to try to do same experiment like this. So here, can you see fish? This is our sterile fish. And here is microinjector. We use this system to transplant cells. In this case, only blue ink you can see here, but you can imagine this is cell. Okay, we can transplant germ cells directly into the gonads of adult fish, like this. Okay, so this is how I can imagine if we can use adult hybrids as a recipient. Okay, this testis of this fish is empty like this, and red cell is donor. Okay, after transplantation, they can, if they can colonize the gonad testis of recipient, they can grow like this, and they can make sperm. This is donor sperm. I want to know if we can do like this. Because if we can use adult, we can get sperm very quickly. And we don't have to wait 6 months. Maybe, in this case, maybe 6 weeks, 7 weeks. After they colonized, they can make a sperm in 6, 7 weeks. It's very short. Okay. Here is donor derived. Okay. Oh, the immunity in the testis also unique. They have blood testis barrier. They can they can make this immuno-free space like this. So I guess donor spermatogonia can survive in the recipient testis. Okay. So here is a urogenital anus is here and head is left, tail is right. Anus is here, and next to right side of anus, you will see urogenic pore and spermatic duct, and we inject the needle into this duct, sperm duct. Okay. Oh, sorry. Here, sorry, this picture is something wrong, but this picture I want to show sperm. But as you see here, after transplantation, donor cells colonized the testis of adult, and they grow and they start to make spermatozoa like this. So in this experiment, donor-derived sperm production was observed at 7 weeks post-transplantation, and 10% of transplanted fish produced motile moving sperm within 25 weeks. So this is a result of the first result of adult transplantation. Okay. So this one is a babies. Okay. Anyway, so this is the last slide I have today. So today I introduced 2 ways of germ cell transplantation in the marine fish. First one, we prepare germ cells from donor testis and transplant into the body cavity of recipient and wait for several months until this fish can get mature. And after mature, we collect egg and sperm of donor-derived gametes. This is one way of germ cell transplantation, surrogate. And another way is this. after we prepare germ cells from testes, we transplant into the testis or ovary of adult fish recipient, because adults, they are already sexually competent. That means ready to make egg and sperm. So they can immediately start to produce egg, donor-derived gametes, egg and sperm after transplantation. Okay, this is the summary of the slide today. And if you are interested in this technique, I'm happy to share the technique or protocols. So thank you very much for listening. Any questions I can?
Yes, thank you so much, Takeuchi Sensei. Very interesting. You introduced two types of surrogate. First, using juvenile, and the second one using adult fish. That's very interesting because it's no need to raise the juvenile, but we can get sperm or egg more quickly, something like that. And I believe there's so many discussion from the floor. Any discussion, you can raise your hands up, or you can write your question in the chat room. Yeah, it is also very interesting. I have some question that how the germ cell can walk to the right organ actually, Sensei. But you already explained that they have a pseudo
Pods.
Yes, pseudopod that they can walk. So actually,
Yes.
the germ cell can walk on the right organ, or they can also walk to the other organ, Sensei.
Yes, they use some signals. I need to explain more about the development of germline. Not transplantation. It's normal fish. Normal fish. The cells, germ cell is very different from other cells. Somatic cells, like the cells can make muscle, skin, eye, liver. They all die when they die. Fishes die. But germ cells can survive to next generation. They are very unique, and their development is also very unique. They are in the very early stages of our development, like cleavage, cell division. Germ cells are established in a different place, not in the gonad. Fish body cells make gonad, but germ cells are waiting when gonad is ready. I think the cells is different animal in our body. Very different. Okay, but they are waiting. The fish can make their gonad, and after that egg embryo make a gonad, the gonad produce the signals, start to invite or show the way the gonad, and the germ cells respond and migrate. But it's only happened in the very early stage of development. In the maybe hatching stage, before hatching stage, egg germ cells in the body is waiting, just waiting. And when the eyes, many other organs are developed, gonads start to produce some signals and then start. So we first need to find when it happened, when they are migrating, moving. So, if we know that, we can transplant. That is a time range, short time range, but we can do transplantation.
Okay. So we need to choose the best time for transplantation, something like that, Sensei.
Yes.
How about the adult? In the adult?
Adult, they don't need, because we can transplant directly into the testis.
Okay.
So inside the testis.
All right. In case of juvenile, just in body cavity, something like that. But in case of adult, you can directly to to the
Yeah.
to the gonad.
Yeah, when 20 years ago, when I was a student, I'm trying to transplant these cells into the gonad of this fish, but couldn't because too small.
Yeah.
Fish is too small. I cannot find their gonad. So I just transplant in near the gonad
Yes.
and expect they show their migration.
Yeah, they
Really.
can migrate. Yes.
Mhm.
Sensei, we have a question from the audience on the chat room from Mas Muhammad Imam Rifai Sang.
Okay.
The question is all that developing gene still safe to the environment, or it just for research purpose only? On the chat room, Sensei.
Yes, I see.
Yes.
Development gene will still save to the environment, or just for research purpose only. It means the question means the fish we make this method.
Yeah, I guess so. Is it the fish, the surrogate fish, Mas Muhammad Imam Rifai? Do you mean the surrogate fish?
Surrogate technique for research purpose only.
Yeah. Oh yes. Maybe how about the recipients, Sensei? Are they can survive in the environment?
Recipient.
Yeah, recipient.
No, recipient. I never release this fish into the wild. We only keep this in a fish tank.
I see.
So yes, for the primary objective of this experiment is make more efficient, effective, efficient, or easy production of aquaculture target species. So we want to use this technique for aquaculture, seed production. And after that, someone have idea, we want to use this for the conservation of genetic resources. So in that case, maybe in the future, in their goal is to release the fish we made from this technique will be released to the wild environment. So could have some problems where we need to change rules or some. We need to be more careful when we release this fish to the wild,
Yeah.
because sometimes the sex is very some sex chromosome is something not natural, because we can make egg with Y chromosome that can make a super male. This is not natural and good for aquaculture, but for nature maybe will cause some problem. Fish super male.
Yes. Thank you for the answer. I open for the other question from the floor. Oh, there is some question from Mas Fitrianto Sang. Is this same technique with the glowfish tetra?
Glowfish?
Glowfish. Glowfish is
Transgenic?
transgenic, I think. Yeah.
Oh, I see. Last week I see in Taiwan they sell in the market, but it's not allowed in Japan. But I was very surprised. Like, glowfish is, I think they are gene transfer, I think. So this is genetic modification is one of the marine biotechnology, but that is different from ours. Our technique is transplantation cell. We only handle cells, not gene.
Yes, thank you. So this Takeuchi Sensei study in transplantation of the cell, but in case of transgenic, in case of glowfish is a transgenic, there is a GFP or RFP or YFP inserted in the genome of the zebrafish, Mas Fitri. Thank you so much for the question. I open for the other question from the floor. You can raise your hand and ask directly. You can raise your hand and ask directly, or you can put your question on the chat room.
Okay, maybe
Yes, Sensei.
I'd like to introduce one of my alumni,
Yes, sure, Sensei.
Anna-san. Anna-san, can you hear me?
Anna?
Anna, yeah.
Hi.
Hello.
Hello. Konnichiwa, Sensei.
Hi, hi. So can you introduce yourself to the audience?
ちょっと恥ずかしい。
No, no, I want to
Okay, thank you.
introduce you because you are expert of transplantation, right?
No, you are more expert actually. You're my senpai.
No, no. If there's someone here have some more questions, you can ask to Anna-san. She got a PhD in Professor Yoshizaki's lab, right? So you use bittering, right?
Yep, I use
Freshwater,
bittering, yeah.
freshwater fish germ cell transplantation. Yes.
Yeah.
Okay.
Okay. Hello everyone. My name is Anna Octavera.
Hello, Mbak
Yes.
Anna.
Takeuchi Sensei said that I was one of alumni from Tokyo University of Marine Science Technology, graduate student from Professor Goro Yoshizaki, study same thing with Takeuchi Sensei, germ cell transplantation, and my phase in bitterling. And yeah, I finished same species, different genus transplantation in bitterling, and we got surrogate broodstock for both sperm and egg. So now I'm working in Badan Riset and Innovation Nasional, the Agency of Research and Innovation of Indonesia. So yeah, I'm just quite surprised when I saw the poster of Yutaka Takeuchi Sensei will give some speech in this webinar. So I kindly to join this webinar. Thank you, Sensei.
Thank you, Mbak Anna. The same institution with Dr. Priadi Setyawan.
Okay.
Yeah, nice to meet you, Mbak Anna.
Nice to meet you.
Yeah. I open other question from the floor. Any other question? Oh, we have question from the audience, Sensei, in the chat room. From Mas Rohmat Rofik. Is this technique can be implemented to all kind of species, freshwater, brackish, and also marine?
Yeah, maybe. Oh, now yes, but if you want to choose freshwater fish as donor, you should use freshwater fish as a recipient, because after they produce egg can survive in the freshwater. Or the egg size, sperm size, maybe egg size. You know, some fish can produce floating egg. Some fish produce sinking egg. We need to choose which donor is, which recipient is suitable for which donor. Second.
Yeah, thank you, Sensei. Other question from Mas Fajar Maulana. In the second method is very interesting. Is there any information or report about endocrine change when the hybrid being maturity in the sperm? Being maturity of the sperm?
Yeah, interesting. Very, very nice question. I like it. But I didn't study about the hormones, but the hybrid fish, they don't have germ cells, but their brain hormone system is normal, I guess. Only if there are some brain, pituitary, and gonad feedback to brain. If gonad is empty, the hormone system is stopped. But once they have some germ cells in the gonad, I think the hormone axis, stream, is start moving, and I think they have that hybrid fish still have our potency ability to produce hormones.
Yes, thank you, Sensei. Committee, do we still have time for discussion, committee? Mbak Sulis, do we still have time for discussion?
Yeah.
Okay. Okay, any other discussion from the floor? You can raise your hand up or you can put your question on the chat room. Oh, we have question from Danang Haryojati in chat room, Sensei. Can this technique work? Yeah, Prof. Kapi.
Yeah, someone tried, but I don't know but that could be very interesting. So, but the problem is the timing of transplantation. We can access to the embryo lifetime. You know, as I told you before, the transplantation time is limited. If
Yes.
we do, like, we need to use guppy babies first. We can wait until they give a birth. I mean, yeah, we need to do some histology work, and we will find the time. But it could be. And I'm also interested in this technique for the sex reversed fish. I know donor cell can change sex in the gonad of recipient. I know, protandrous fish, progynous fish.
Yes, thank you so much, Sensei, for the answer. I believe there's still many discussion from the floor, but because we have a limited time, so thank you so much for the presentation and thank you so much for the discussion from the audience. Please give big applause for Takeuchi Sensei.
Thank you.
Thank you so much, Sensei.
Thank you, Sensei.
We do hope we can have a collaboration in the future.
Yes, of course.
Yes.
Thank you so much.
So I will close my screen.
Yes, thank you so much. And we move to the next presenter. The next presenter will be from Dr. Priadi Setyawan. Dr. Priadi Setyawan will talk about recent updates of tilapia breeding program in Indonesia. Before Dr. Priadi Setyawan present his talk, I will introduce Dr. Priadi Setyawan first. Dr. Priadi Setyawan get PhD from Department of Fisheries Gajah Mada University, and he get his PhD from Animal Breeding and Genomic Department of Animal Sciences Wageningen University and Research, the Netherlands. Dr. Priadi Setyawan has so many good paper in aquaculture and also in some other journal. So Dr. Priadi Setyawan now is a researcher at the Research Center for Applied Zoology, National Research and Innovation Agency, or BRIN. So Dr. Priadi Setyawan, are you ready? I think you do. Time is yours.
Thank you for the time, Dr. Tini.
Yeah.
You see my screen?
Yes. Could you please,
Sorry?
Full screen, please.
Oh, sorry. Let me check the...
Yes. Thank you so much.
Hey, good afternoon, Professor Alim Isnan Setio, Prof. Yutaka Takeuchi, and also all webinar participants. So today, I would like to share about the recent update of tilapia breeding in Indonesia. So first of all, I would like to introduce the tilapia and aquaculture in Indonesia. So why do we need tilapia? So as part of the food security program in the future, we know that global population has exploded about 3 times in the mid-20th century. And for now, is the population of the world population is about 2.1 billion in 2023, last year. And it's predicted that in 2050, the number will rise until 9.9 billion. This is a very big number. We can see from the right side. Yeah, can you see my pointer?
Yes, we can see your pointer clearly. You can use your pointer, I think.
Okay. Yeah, here we can see that China and India is in the top list of global population. But we also can see that Indonesia also have a significant increase. And for now, the number is about 274. It's also a big number. And this brings implication to the future food security is kind of a challenge for the future. And the global protein demand in 2050 will also is predicted increase to about 52% in 2050. So the main source of the animal protein are from livestock and from poultry and also from fish. As interesting that for fish, according to FAO 2022, that fish supports about 17% of the global animal protein. So I think this is very familiar figure. That fish is mainly produced from 2 sources, from marine water in capture fisheries and also from aquaculture. In capture fisheries, we can see that consists of capture fisheries in marine waters and also capture fisheries and inland waters. And the rise of global capture fisheries from about 1990 to 2018 is about 14% or relatively stagnant if we see in this picture. But if we see the aquaculture industry from 1990 to 2018, this rose significantly. And this comes from aquaculture from marine waters and also aquaculture in inland waters, with a very big number. The increasing is about 527%. So aquaculture industry is very promising in this. According to FAO, the aquaculture will have a significant contribution to food security in the future. And this industry is the most efficient protein producer. So in this example is a salmon industry. That's the production is like this. So the FCR, feed conversion ratio, is relatively low. And the highest is in livestock, is about 6 times larger than fish, and also half of the poultry. And of course, its water consumption is quite low. And the most important, we can see also in the picture in the left, bottom left, that's the CO2 equivalent emission per gram of protein source is very low in compared to other protein source. This is very important for the future because we are more green. And now Indonesia is the world's second largest fish production. We can see here that the production of Indonesian aquaculture is second after China. It's about 13 million tons. but is far behind China. In China, we can see that the rise starts from 1990 and until now can reach up to 70 million ton. It's a very big number. The interesting thing that in Indonesia, there was also a significant increase since 2000 and I believe in 2011 here. It's marked in a very small rectangle, green rectangle here. That's I remember in this period in 2011 to 2014, there was a policy by the Ministry of Marine Affairs and Fisheries to push fish production up to 353% between these periods. And this was there was also a Broodstock Center in Sukamandi that organized Broodstock meeting annually, and also there was a annual meeting for breeders, also for institutions who also conduct a breeding program. That's we also have update for each single activity in their facility. However, since I think in 2014, there was a different policy to focus on marine activities and in particular to protect our fish from illegal fishing. And I think it seems that the policy has significant effect on the reduction of national aquaculture production. Here is, we can see the top 5 of aquaculture species in Indonesia. Nile tilapia is in the top position and followed by catfish, shrimp, milkfish, and carp. Tilapia is commonly farmed in these environments and normally in earthen pond, in concrete pond, in rice fish system in combination with rice. And a major part is produced in freshwater floating cages. And also that's this dominates the tilapia production is about 95%. And also a small part of tilapia production was produced in brackish water ponds in an expensive system, which the density is very low. It's about 3 to 5 fish per meter square, or even less if the farmers apply super extensive. I mean, they stock their fish without foods, only rely on the natural foods, then they can stock 2 meter with 1 tilapia. And also another practice in polyculture with shrimp like this. And in this scheme, if in particular, if they provide also the aeration, they can increase the density of tilapia up to 5 fish per meter square. And we also tried our tilapia in marine cages in Seribu Islands. And in these experiments, we can produce up to 1 kg in 9 to 10 months. However, the survival rate is was quite low. So it's also our challenge to produce more tolerant tilapia. Now we go to the tilapia breeding program in Indonesia. We can see in the right side is the list of tilapia that has been released and also that's available in the markets in Indonesia. Majority they came from they developed from gift that's was imported in Balik Kanwar in Subang in 1994. Then because of the it's very easy to be farmed and then spreads widely to other regencies. And then since then the breeding program for tilapia is very developed very fast. we can see here in the first in Jambi, in Jaika in 2004, and then Nirwana in Purwakarta in Java. Is most of the breeding program stated in Java. So in here, Nirwana in Kesit, we can see here in this area also in Sukabumi, also another strain that's been released. We also have the major tilapia is red tilapia, black tilapia, and also there's a combination between red and tilapia. It's normally a hybrid between red and black tilapia, but the color is a specific location. For example, the farmers in West Java, they tend to they prefer to farm black tilapia, and the farmers in Central Java, in Semarang, in Yogyakarta, they prefer to farm red tilapia due to the different color. So how do we produce tilapia? We have a kind of a national protocol for this. We call it National Protocol for Tilapia Breeding Program. So this protocol is based on mass selection and also family selection, and also some of breeding institution we perform hybridization. So this selection based on phenotypic selection, mostly in body weights. So how do we select fish? We have a population in the pond like this, and then we select the top of the performance, for example, for body weights. And then we use this as a broodstock, and this is the results. The progeny is not the same with the parent, but slightly lower, and this what we call is in breeding program is a response to selection. For more detail about the breeding scheme can be seen in the right side here. We have a national protocol that's required at least 30 families. So families is a combination progeny. It's a progeny resulted from one male and one female. So this can be produced in 1-by-1 mating combination. Normally we use 5 hapas. So for family one, and then we can get about 500 fry is normal for tilapia, or more or less is 500. And then we can select randomly for 300 fry per cage, and we grow in earthen pond for until we can differentiate the sexuality between male and female, and about 150 we can grow separately in different hapa between male and female, and in the end we have like the mean of the population of male and also mean of population of females. Then we select the best 10% of each cage. Then we can use for detect this fish, and we can use to produce the next generation for the next generation. And the selection response for this is normally is about 10%. We also produce saline tilapia from brackish water ponds. So here we have to differ to strains specifically for brackish water environments, which is Srikandi strain and Salina strain. So this is the ministerial decree that we obtain when we release a new strain to the farmers. So this strain need to meet the requirements, like for several tests to proof that we produce a new superior broodstock that's different from the previous. In the right side is the example. Here is the example of the certificate. So this two type is Srikandi strain in 2012, and this is Salina strain in 2014, specifically for brackish water environment. Our second strain was produced by selection and also continued by hybridization. So selection was made on this fish in aureus, and we use male of this species, and then we combine with female of Nirvana strain that's has been produced in Purwakarta. And then we combine, and then this is what we call Sikandi strain. Normally, the combination, if we refer to publication, that's the combination of aureus male and Nile tilapia female will produce male dominant population that is also is part of sexual dimorphism, and it is more preferred by farmers. But in our case, our Sikani strain produce more or less similar. It's about 60% of male and 40% females. And now we later on we know that our aureus is not pure aureus, but already Sikandi actually. So we knew that our what we call aureus, now we call it Sukamani strain, has been integrated by about 8.6 genome of blue tilapia. And we found also 5 genes that's related to salinity resistance are included integrated from aureus. So it's kind of a unique population that we have in our in Research Institute for Fish Feeding in Sukamandi. And this is has been published in Aquaculture in 2022. So how about the future prospect of tilapia farming? I think this is about saline tilapia. We are in is an archipelago consisted of more than 17,000 islands, and also because of many islands that we have the second longest coastline is about 91,000 kilometers. And of course, this is a high potential area for brackish water aquaculture. That's the opportunity, the potential area to develop is about more than 6 times than freshwater. So we develop a precision breeding program for Sukamandi strain that can be farmed in these environments. So this is our project funded by Coupon Stichting in 2018 and 2023 to produce more resilient tilapia using Sukamandi strains for this environment. So this is John Pensy, Professor John Pensy from WorldFish, and this is Mark van der Poot from French National Agriculture. And this is my supervisor, Mark from NIWA, New Zealand. And this is my professor, is Hans Komen from Animal Breeding and Genomics. So we had a discussion of this program. And what about the status of our tilapia in Indonesia? From all these strains that has been released, we only have 2 strains specifically for brackish water. And our survey in 2020 that has been published also in Aquaculture 2020, that a majority of brackish water farmers, they use black tilapia freshwater, like in Central Java and East Java, in West Java, and also in Banten. Only a small part that use saline tilapia, and a majority of them use black tilapia than red tilapia. So for the reason is that if we use red tilapia, is kind of attract predator in the specific environment in brackish water, which is normally open, quite open without vegetation. That's make the survival rate is very low. So this is the distribution of tilapia used by the farmers. Most of them use tilapia in polyculture system, especially in East Java, and also we can see also a major part in Central Java, and more here we can see that polyrotational is also preferred. And in Banten is kind of specific that they use tilapia in monoculture. They shifted shrimp farming to tilapia farming because of highest impact of mortality in shrimp. So in our project, we improve the selection using blood methods. So here, the selection is our national protocol based on the phenotypic selection. This is an example of the protocol. But there is some issue here. That's about what about the desired traits of the species that we are going to select, and what kind of
Excuse me, Pak Aswar, could you please mute your microphone, Pak Aswar? Our committee, could you please mute Pak Aswar? Yeah, Dr. Priadi, you can continue.
Yeah, I'm sorry, I cannot see the other participants, so I just okay, I will continue. So some issue with the previous is that what about the genetic parameters? Can we precisely predict the genetic parameters? And what about the inbreeding rate per generation? And also, what about the accuracy of selection? And some of a majority of the breeding program in Indonesia is they stop to operate, they discontinue after 4 generations. So it's also a big question about the sustainability of the breeding program, why they stopped the breeding program. And some of them, they said that the variation is lower, and it means that response to selection is also lower in the after 4 generation. And also that's how we have a breeding goal for multiple traits can be also obtained together in these methods. So for this, we develop using GBLAB or base linear unbiased prediction. And here is based on the assumption that we select the additive genotype rather than phenotypic, because if we select the phenotypic, then also part of the selection is the environment. So this is the basic formula of the plot. So this is the metric for fixed effects, and this is the metric for random effect that we are going to predict. And we know that this is the blue and black. Yeah, blue is base linear unbiased estimates that are solution or estimates associated with the fixed effects. Fixed effect is some of environmental that can affect to the sorry, yeah, to the phenotype. And BLUB or base linear unbiased prediction are the solutions, but identified as a prediction associated with the random effect of our model. So this is the normal model. So in first, we can have a solution for this. We can have a solution for blob, and we can also have a solution for blue. So this is the basic scheme of our breeding program. So we have the scheme. First of all, we need to define the production system, and then what kind of breeding goal formulation, what kind of desired traits that's chosen by the farmers, by the market, and then how do we collect the information, the phenotypic. We collect the family relationship using PIT tag or genotypes. Then we need to define this, and then the selection criteria that we mentioned before about the genetic model, and then how do we select fish for the next generation using a breeding goal. Here we also perform an experiment using lab to check whether our nucleus, our facility breeding program is similar or not with the commercial farming environments in brackish water. For this, we conducted an experiment in freshwater in Research Institute for Fish Breeding, and also we conducted the test environment in brackish water farmers. For this, we use G5 generation 5 of Skamani strain. The substantial GPIE between two environments is about 0.6 for DGC, for daily growth coefficient. It means that we need to incorporate the information of the production environment to our model in order to have accurate prediction. If not, then our selection will perform differently in commercial farming environments. Or we can say that if we select fish in our facility, and then if we not incorporate with the information from brackish water, then they can perform differently. This also has been published in aquaculture about the GPIE, genetic parameters and genetic bioenvironment interaction between freshwater and brackish water. Then we also perform this experiment. So we collected all information from the farmers. For this, we involved 224 farmers from 4 provinces in 7 regencies. So we developed questionnaire, and then we use pair-based comparison matrix like this, and then we define the most preferred traits by the farmers using analytical hierarchy process. And we found that growth still in the top list, and the salinity resistance, survival rates, disease resistant, and RCR. This has been selected by the farmers. So what the point of this? We can use this to select fish and to to define the breeding goal, what kind of direction that we will use for our breeding program. Here's actually the main differences between our national protocol and lab protocol. So in our national protocol, we select phenotype using this the previous mentioned for a scheme that's based on the trait that can be measured directly in the farm. And we select the top 10, the best performance of its population. And when we use lab, it means that we select genotype. It means that this selection can be transmitted to the next generation. because we also correct the environmental effect here. So this is again the basic formula. So the response is kind of the combination of the fixed effects and the random effects and also the residuals. So the main differences for the selection criteria for PPEN is based on phenotypic selection, and for BLUP is genotype selection. And the calculation of inbreeding rates for PPEN is based on assumption. For the assumption, it means that if we can produce more than 30 families, that is sufficient to have a selection response, but also respect to the inbreeding rates. But I think based on the reference, that's some references that's mentioned that the minimum number of family is about 50, because then if about 50, then we have inbreeding rates is more or less 1 or in acceptable level. But if we use BLUP, we can estimate this in a software. So it's more, it's kind of a prediction that's more accurate. And we also can measure accuracy and bias in these methods. The other difference is on genetic gains. That's in PPN is kind of maximum selection. We can select more higher intensity, more intense. We can select maximum. But in BLUP, because we normally use optimum contribution selection, or we can use Fishboost software for that. And we take a balance between response to selection and inbreeding rates. So this kind of optimum genetic gain that we obtain. This have a significant impact on the sustainability of the program, because we can produce the next generation with the acceptable level of inbreeding. It means that we can select in the next generation without reducing the variation of the additive genetics. And also here, another difference is traits for our previous protocol normally is based on one single trait. That is most common trait is body weights. But in BLUP, we also can combine to multiple traits like for fillets, for FCR, for other traits that can be measured. So precision breeding using BLUP is more sustainable than phenotypic selection. This is what happened to if we select using phenotypic selection, normally in 3, 4, and 5 generation, and then stop because of we can select more because there is no variation. It means that the selection response will also low under 10 or even 5 or less than 5. But if we maintain inbreeding rate per generation, we can select more to the next generation. And in one point, we can have a fish that grow double than the previous broodstock or even triple, for example, for chicken. And this, what we use in calculation, we use R-based programming. So this is several software that can be used for precision breeding. So selection and MOPS or modular breeding program simulation is when we can define before we start the program. And R is open source program that you can perform any calculation here. And I use RSML-R is a specific software for BLUP that can also handle a big data for this. And another software is we can use for simple, for TMU Wombat LM4 and DWR REML, etc. So we can use many different kind of software. Here is the BLUP selection in our fish in Sukamandi, in Research Institute for Fish Breeding in Sukamandi. So we produce the first to fourth generation using phenotypic selection. At least the minimum requirement is 30 families, but in our case, we even produce less for this. For example, all of this produce less than 30 families due to of the mating problem. We use 1:1 ratio, but it seems that it's not ideal for tilapia, because if male and female, they are not in the same level of reproduction periods, then the male will attach female, or on the other hand, and what we found is that half of our broodstock died during the mating process. And then we introduce 1:3 female in our combination, and we successfully produce 91 families in generation 5, and we also produce 102 families in generation 6. Normally, we produce this in 3 months, but we can produce 102 only in 1 month. So I think it's a good solution for this. So these 2 generations have been produced using club selection in a secure set. So here is in our facility in the Research Institute for Fish Breeding, and this is the brackish water in commercial farming environment. So we also use here is PIPLAB. So this is a PIPLAB method that utilize individual relationship metric to estimate genetic merit of individual of our Sukamandi strain. We can trace back, for example, here, this is the parental line. We can trace back the parental line of each fish that we used in the model. So for this picture, the male line is marked with the red color, and the female line is marked with the orange color here. So the parent contribution to the selection can be managed effectively using what we call is optimum contribution selection, a method by Mewison. That's we can also use like fish boost to have a balance between response to selection for a specific parent, and also the level of inbreeding if we use that fish. So we can manage which one fish that can be used to the next generation. In the right side is the model of the genetic model that we use to perform the 6th generation of our fish. So this content of fixed effects and also random effects, this part. We also included the common environmental effect is also part of the random effects. This fixed effects is initial, that's normally for tilapia, different different age, also different body weights also have different effects to the results, and also for sex, because we know that there is a sexual dimorphism when the male tilapia grow faster than female. Then we also put this as fixed effects. Yeah, in the end, our selection methods resulted in very low bias, like this one. The bias was about 1.4 in brackish water, which can be seen in this picture. This is from the green line is reference line, and the blue one is our prediction. So we can see that this is low bias of estimation. And I think that the future that's saline tilapia is more promising to be developed, because in May 2024, our president also released this program So in the Ministry of Marine Affairs and Fisheries, mentioned that tilapia farming in lakes and reservoir could damage ecosystem and pollute the water, and also due to conflict of interest with other sectors. That's why we try to move our farming habitat from freshwater to brackish water, because we still have a very large, and also that's yeah, you can see like this picture that's in the right side. It's very wide, that is normally found in brackish water. So this can lies from 1 kilometer from the coastline or to 5 kilometers. It depends on the slope of the in the area. So the ministry has developed a strategy to involve to develop salin tilapia in 80 hectares of land in Karawang. And in the future, this will be extrapolated in other regions in Indonesia. Yeah, I think for the conclusion is that the current protocol based on phenotypic selection has successfully produced superior tilapia. However, for sustainability reasons, the total family, the minimum total family needs to be increased up to at least minimum at least 50 families. The current protocol could be improved using blood to increase the accuracy of selection. And then the desired threats and the commercial protection environment need to be defined carefully to avoid what we call its genotype environment interaction. And for future prospect for tilapia farming is in brackish water environments, then the number of salin tilapia strain need to be increased for the future. That was a nice summer in my professor house in Wageningen, and thank you for your attention.
Thank you so much, Dr. Priadi Setyawan, for the presentation. It's very interesting, since we believe that salin tilapia is very potential because we have a second largest coastline, like 90,000 kilometers, that is very potential for a salin tilapia production. So now I open the discussion from the audience. You can put your question on the chat room, or you can ask directly you can ask directly for us. Mic on and came on. Any discussion from audience? So let me start with my own question. So I'm really surprised that tilapia now is the largest product. Fish production, do you mean, Dr. Priadi?
Yes.
Fish production, yeah, not included seaweed, no?
Fish production.
Oh, for fish production. So yeah, there's a very large. Could you please open the slide? Yes, that one. Yeah. The biggest aquaculture product in Indonesia right now, right?
Yes.
So mostly for local market or exported?
Well, I think this based on the total production also include for local and also export markets. This number come from the Marine and Fisheries in figure in 2022. I think it's pretty up to date. And also consists of all tilapia production from brackish water and freshwater.
So far, how about the saline water tilapia in Indonesia?
Well, this saline tilapia is very promising to be developed, but until now, our farmers, based on our research in 2020, the total farmer that use the saline tilapia is only less than 15%, so it's very low.
Less than 15%.
when they use tilapia in their environment, in their farming environments, in brackish water, they normally have very low survival rates. Normally, it's about 30 to 50. If more than 50 is considered successful, but if normally the farmers get very low in it's about 30%.
Until now, still very low, but I think this is very potential, and congratulations that you use a lab method, right? Best linear unbiased predictions for saline tilapia breeding. So congratulations.
Thank you.
Yeah, I think this is very potential to raise up the aquaculture production in the saline water area, I think. Okay, we have a question from Mas Fajar Maulana in the chat room. Mas Priadi, what are your tricks and suggestions if we want to implement this selection principle in Indonesia, especially regarding maintaining the purity of the selection population? Thank you so much.
If you want to implement selection. Well, actually, for blood, one of the requirement is we use attacking. So each fish that can be, I mean, as a selection candidate for the next generation, each single fish need to be tagged. So for this, we use PIT tag. So we call it passive integrated circuit or transponder, I mean. So it's kind of a microchip that we insert into the body of fish. So, for example, in the upper, when fish reach the size of 5 grams per fish, so we can inject with this microchip, and it means that we can define which fish and which, and then we combine this information, this tag number, with the data in our system, in our computer. So I think that the purity we can maintain as long as we the tagging still inside the fish.
So using tagging method, right?
Yeah, we use a microchip. That's in the past we used a tag number. So we use a plastic tag contain a specific number, and then we tied into the caudal. But then during the grow outs, sometimes the tags lost. So we use microchip for this. We inserted a microchip that's similar that we use for dog, for cats. Is content of information of the number of specific number that is not very specific, so it's not double with other tag.
Yes, any further discussion from Mas Fajar? Oh, we have question from Professor Takeuchi Sensei. Thanks for a great presentation. In Japan, fish genetic breeding researchers are interested in creating high water temperature resistant strain, global warming, and vegetarian fish strain, low fish meal feed. Oh.
Yes, thank you. My question is that in the future, maybe we need more fish strain to adopt high water temperature and low fish meal food. Is there anything you try to do right now? Or maybe you're focusing on growth?
Okay, thank you for your question, Professor I think that this most important for the future. For the future is also include the survival. In this case, is the what we call it is to respect to the environments is the global warming. We need to also measure the temperature resistance. So there is a method that we use is we select fish that can survive in what we say is a abnormal situation. So for example, when the temperature goes down or up especially during the rainy season, that's the temperature with it's reduced significantly, then the survive is also can be used as part of the next selection. And also for the high temperature, because temperature also increased significantly. And I think this is also in our concern. But I think for the vegetarian fish, for tilapia, we didn't use this in our part of our selection. But we know that tilapia is kind of omnivorous fish, and in brackish water, normally they eat phytoplankton. So it's kind of strategy by the farmers to obtain the RCR below 1, because they stock commercial, very low commercial feeds, and they rely on the natural foods. And they use fertilize to combine with the commercial feeds. But it's also, I think this is very nice if we can also include it in our in our future selection program.
Thank you.
Thank you so much for the discussion. We have another question on the chat room from Mas Agung Prastio. Is there any research in tilapia breeding who focuses on increase the fillet yield? I saw in your slide, if fillet yield it isn't in the desired rates. As I know, the frozen fillet price in USD 6.7 something kg per kg and 19,000 something per kg. It means that the price is 4 times higher than we sell in local market with whole size. In my calculation, if we can increase the fillet yield just for 1%, we can increase the profit, and it means a lot for our processing company and tilapia farmer in Indonesia. Thank you so much for the discussion, Mas Agung Prasetyo.
Yeah, thank you for your question, Mas Agung. It was really a good point, I think. Yeah, fillet is very important. But actually, our project was about how to help small-scale farmers in brackish water that consists of 65%. So fillet yield is not really, it's not well known by the farmers. So it's not part of their desired rates. But for in our case, in brackish water, that's the harvest size is about 200 and 250 and sometimes in 300 gram per fish. That is very low. It's not sufficient for fillet yield. But I think for fillet yield can be included in a selection program if we grown fish in, for example, in the lakes, when our target is for fillet up to 700 gram or is 1 kg, then we have to include selection for fillets. I think that makes sense. But for very small, we can measure for body depth. It's also part of how many proportion of carcass yield in this scheme.
Thank you, Dr. Priadi. Any further discussion from Mas Agung? We have another question from Dr. Rinda. How long does it take to do fish selection by using the blob? And the second question from Mbak Mary. What a great presentation, Dr. Priadi. I want to discuss about the multiple trait in blob method. In your experiences, how you measure or maintain the multiple traits, And the last question from Mas Santoso. The biggest challenge for saline tilapia is its economic potential compared to other fisheries commodities such as shrimp, but polyculture can be introduced more as a sustainable cultivation system. Genetic achievement for saline tilapia, I think it will answer the potential production. Okay, there is discussion from Sri audience. Could you please respond for that, Dr. Priadi? Thank you.
First, I would like to answer the first question from Dr. Indah about the selection process. For plot, I think the process is similar with previous method because we also use the same environment, we use the same aids, also the same PCP, the same harvest size, but the difference is the technique that we use. That's how we manage, how we evaluate, how we predict the breeding value of its animal. That's the difference. So in terms of time, I think that's pretty similar. Using PLAP and also using the previous methods. Considering the what we can say is the generation interval for tilapia is 1 year or less, so we can select fish is less than I believe in 7 months we can produce a new generation using BLAP method. For the next question. Well, for the multiple threads, I think that's the multiple threads is based on the desired threads by the farmers, for example, for body weights and for FCR. So we have an estimation of on breeding value of its threads. So for example, this species, we have a breeding value of growth, and then we have breeding value for in this case is FCR, and also breeding value for other traits. And then we can combine together in what we call is breeding goal. Breeding goal is a function between the breeding value times the weighting, the weighting, what you call it is that's can express how important is the traits. So we can make a scale, or we can use economic value of its traits, and then we can combine in one breeding goal. So that's we select fish. So in one direction we can select a multiple traits in one fish. It could be one fish is not the maximum, the fastest grow, but the best fish based on the breeding goal that we that we select based on the preference. Is that answer the question?
Any further discussion from Mbak Mary? So you can continue for the next.
For the next question from Santoso. Yeah, I agree that the polyculture is more preferred, and also will polyculture is a more sustainable cultivation system. And also this system, polyculture, has been practiced by the farmers in many in many agencies. Yeah, I like to show the result for this. Can you see my screen?
Yes.
Stop to. Yeah, actually, polyculture is most preferred farming system by the farmers so far. because they try to develop shrimp, monoculture shrimp, but also they found a problem with diseases. And when we grow only tilapia, then the profit is not so high as so in terms of sustainability, also in terms of profitability, polyculture is the best, I think.
Okay.
So for the next question.
Yeah, I think Mas Rahmat Rafiq raised hand. Maybe you can ask directly.
Yeah.
Yeah.
Thank you, Pak Priadi. Thank you. I'm Rafiq from the Directorate General of Aquaculture. As you know that we have a program, the aquaculture of saline tilapia in Karawang in our facility that already visited by our president, and the next step is that we want to expand this program to culture the saline tilapia to all ponds, shrimp pond that under development. So, what do you think about how to develop the saline tilapia, since the program will need more seed, more tilapia, saline tilapia seed, and also more
I'm sorry, Mas Rahmat Rafiq, maybe due to internet problem, we cannot hear your voice. I think there is some internet problem. So sorry, Dr. Priadi, can you catch up the question? Oh, can you catch up the question?
I get the point that indeed in the future we will need more seed for freshwater farming. That's as our government has a policy to develop more tilapia in freshwater. So the bottleneck is about the fry production. As we know that tilapia is, I think, if we compare to another other species like catfish or carp or milkfish, pangasius, the number of fry or the fecundity is very low. I think this is indeed is a bottleneck in terms of production capacity of seed. But if we want to increase the ability to grow more in brackish water, then we need to continue our breeding program. And we can increase the selection in higher environments, like for example, in the salinity more than 25 ppt. Like we know that our farmers, they use more tilapia in combination with shrimp in a polyculture system. So we propose to perform a selection under the same condition with shrimp, at least in between 25 to 30. I think that's the question, I think.
Yeah, thank you so much, Mas Priadi, Setyawan. I believe there's still many questions from the chat room, but because our time is up, I think you can directly ask to Mas Priadi for the next the other question in the chat room. Is it okay for them to message you directly, Mas Priadi?
Yeah, that's fine.
Yeah, that's fine. So thank you so much for all the participants. Thank you so much for all the keynote speaker for sharing your valuable material today. Thank you so much, Mas Priadi. That's for our SinnTech webinar, the 7th SinnTech webinar today. Again, thank you so much for Prof. Yutaka Takeuchi and also Dr. Priadi Setyawan. So I think the committee will send, if you don't mind, could you please send your battery, and because some audience asked for the material as you present today. So please, the committee will send to you for the participant who wants the material, I think. So thank you so much. I return it back to the committee, Mbak Sulis, or from the department.
Me.
Okay, Mbak Sulis. Thank you so much.
Yes. Thank you, Dr. Dini, for guiding the webinar. Thank you very much for Professor Yutaka and also Dr. Priadi for such an informative and engaging presentation. We appreciate your time and effort. And then, ladies and gentlemen, now we would like to take a moment to recognize and honor our speakers with certificate of appreciation. I would like to invite Dr. Eko Stiobudi to come forward and present the certificate to Dr. Priadi.
Okay, thank you, Mbak Sulis. First for Prof. Takeuchi or to Pak Priadi?
Yes, maybe Takeuchi Sensei first, but maybe Takeuchi Sensei already leave because
Okay.
we have
Okay, thank you.
another appointment.
Thank you. Pak Priadi, thank you very much. Teacher department really appreciate your presentation, and now we present certificate for you, and hopefully it's useful for you. Once again, thank you very much. Thank you very much, Pak Priadi.
Thank you, sir.
Okay. Okay. Okay, Mbak Sulis said it's okay.
Thank you, Dr. Eko Stiobudi. Okay, ladies and gentlemen, for information, the certificate for participant will be sent by email. And then to all participants, don't forget to fill the attendance link in the chat box now. And before we close this webinar, we will take the picture. So to all participants, please open your camera. There are several slides. So for the first slides, open your camera and then 1, 2, 3. For second slide, 1, 2, 3. 3 slides, 1, 2, 3. 4 slides, 1, 2, 3. And then 5 slides, 1, 2, 3. And then 6 slides, 1, 2, 3. 7 slides, 1, 2, 3. And then the last one, 1, 2, 3. Okay, ladies and gentlemen, that concludes our webinar for today. We hope you found it valuable and insightful. On behalf of the organizing committee, I would like to thank all our speakers and also participants for their contribution and presence. We look forward to seeing you at our future webinar at SinnTech 18. That's all from me. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih rawuhnya, matur nuwun sangat
Terima kasih.
sharingnya.
Thank you sangat.
Terima kasih Bapak Ibu.
Terima kasih.
Ya. Mm.